Hello again, everyone. We have a long and incredibly enjoyable afternoon of microbiology ahead of us today. But before we start, really, is the results of round one of microbiology microbe of the week. So some answers. Thank you for everybody who sent me in some answers. Everyone else, it's not too late. Look, they've only got like 2 points up here. Anyone who wants to join, you can easily catch them up. So the question was the following. So why is it that mycobacteria are naturally resistant to beta-lactams and vancomycin, even though they have peptidoglycan? So it's something to do with the way the cell wall is organized. So gram-positives, they're like this, okay? Plasma membrane, big, thick layer of peptidoglycan. So beta-lactams and vancomycin can diffuse across here and inhibit the enzymes involved in peptidoglycan synthesis. Gram-negatives. Outer cell membrane with porins. So the beta-lactams can get through the porins, but vancomycin can't. The question is, what is special, or the idea of the question is, what's special about mycobacterial cell walls that makes them naturally resistant to both these classes of antibiotics? So they have a peptidoglycan layer, and they have an external membrane. But the constitution of this external membrane is very different chemically from what you find in gram-negatives. It's got mycolic acids, and instead of having LPS on the surface here, they've got lipoarabinolactams to give a kind of different structure. So the lipid part is very long chain, which means that the consistency of this membrane here is not like olive oil or butter, it's like wax. So it's very, very difficult for hydrophobic antibiotics to diffuse across the membrane. And the membrane porins, which there are, of course, have a completely different permeability compared to porins in gram-negative outer cell membranes. So that means that, in fact, the outer cell membrane of mycobacteria, like Mycobacterium leprae and Mycobacterium tuberculosis, it's impermeable to beta-lactams, vancomycin, and a lot of other antibiotics. So that's why if you've got tuberculosis, you can't just go and get some ampicillin from your doctor. You need to have a long, yeah. Okay, you've got plasma membrane, peptidoglycan layer, and an outer membrane. But the chemical composition of this outer membrane is very different and very impermeable to most antibiotics. Also something we'll see later, mycobacteria phylogenetically are part of the gram-positive family, okay? So it's almost like they've evolved an outer membrane independently with completely different chemistry. So that's why the permeability characteristics of this membrane are very, very different. Okay, very difficult to treat mycobacterial infections with antibiotics. You need a cocktail, and you need to take them for a long time to be cured. Okay, so that was that question. So yeah. Oh yeah, second part. What's the relationship with Scarlett O'Hara? Well, several people found the answer. I guess it's probably on the Wikipedia. Yeah. Yes. Yeah. The actress who played her was killed by tuberculosis. Yeah. Yeah. Not the only great artist killed by Mycobacterium tuberculosis. It was known as like the White Plague in the 19th century. You know, Frédéric Chopin, the famous victim. Yeah, it was like the AIDS of the 19th century. What else to say about this? Yeah, okay, so can we move on now? um, yeah, so the other day we got up to this part. We looked at cell wall structure, peptidoglycan synthesis. So the objective for the next hour, hour and a quarter, is to go through all of the other structures of the bacterial cell. Starting with, okay, well, what is left of the cell wall in gram negatives? Lipopolysaccharide, because we didn't get up to that yet. So never forget, lipopolysaccharide is an essential component of the gram-negative cell wall, and its structure is like this. So, okay, lipo, it's got a lipid part, and then polysaccharide. So the lipid part is called lipid A, and then the polysaccharide part is composed of a core oligosaccharide, which is highly conserved among gram negatives. And then after this, you have tens, dozens of repeats of, well, a particular oligosaccharide. So, and this is variable between bacterial strains. So this is what's known as the O antigen. This little diagram here is not really fantastically good because you don't have a branch structure. It's really kind of linear. Each repeating subunit is going to be attached one after the other. So what is the function of this? Well, it protects the outer membrane from chemical attack, from detergents, bile salts, for bacteria that live in the gut. They have to survive passing through the small intestine, where you have a whole load of salts, bile salts and choline that's secreted to disrupt fatty acids and membranes. So LPS can protect bacteria from chemical attack, and also against kind of accessibility of the outer membrane to some kind of antibiotics. Now, this part of LPS, lipid A and the core oligosaccharide, it's highly conserved, and it's kind of on the outside of the bacterium, and it's recognized by receptors of the innate immune system, which tell our immune system that we've been infected by gram-negative bacteria. So once LPS is present, if you've got an infection by, I don't know, salmonella or a nasty E. coli infection, then the LPS on the surface of these bacteria is going to be detected by different innate immune receptors. And this is going to start up inflammation. Locally, this is good. This is a positive thing, right? It helps you resolve the infection. However, if you've got septicemia and a large number of bacteria in your blood, then this is going to cause a big problem, because you'll have endotoxin, LPS, all through your body, and you'll have a systemic activation of your immune system. And this is what causes toxic shock. So, fever, septic shock. So, yeah. Okay, so the last thing is if you have a systemic infection, if you have septicemia, bacteria in your blood, then you'll have activation of a whole of systemic activation of your immune system all through the body, and this is very dangerous. That's what gives you septic shock, and you can die. And it's mostly down to the presence of LPS, endotoxin, which hyperactivates the immune system. So medically, this is very important, LPS, but that's not why bacteria have LPS, okay? One of the things that is kind of the wrong way to understand this is thinking, okay, LPS, it's endotoxin, it's there to give you septic shock, okay? That's not why bacteria have this, okay? All gram-negative bacteria have got LPS, and most of them are not pathogens. So this is why they have it, okay? And our innate immune system can recognize that because this core structure is very highly conserved. Okay, so the conserved part activates your immune system, and the variable part, this is different between bacterial species, and within a bacterial species, it's different between strains. So it can be used diagnostically to identify different strains of bacteria. So this is called the O antigen, O for O's, okay, it's made out of sugar. Now, how is LPS synthesized? It's the same problem as for the synthesis of peptidoglycan. It's a complex polymer. It's got to be exported outside the cell. But there's no source of metabolic energy outside the cell. So it's the same problem, and bacteria are going to use the same solution to synthesize this polymer. So on the one hand, you've got synthesis of the core oligosaccharide, which is synthesized on lipidae on the cytoplasmic face of the plasma membrane. And the whole thing is flipped over into the periplasm. On the other hand, you have the O antigen repeats, which are also synthesized on the cytoplasmic face of the plasma membrane, and again attached to a lipid transporter. And this is also going to be flipped over in the membrane to transport the repeating subunit into the periplasm. Then you've got a transglycosylation reaction, which adds this subunit onto an elongating chain. And this repeats for several cycles until, I don't know, the bacteria kind of think is long enough. And then this whole chain is going to be transferred over onto the core antigen. This step and this step, like the synthesis of peptidoglycan. So what do you think this could be? Come on, it's a leading question. I wouldn't ask you if it was impossible to find the answer from what you already know. What was this in the synthesis of peptidoglycan? The transporter. Bactoprenol, right? Yeah. Bactoprenol diphosphate. It's the same thing. Okay? So the repeating subunit is synthesized on the bactoprenol diphosphate, flipped over, and then the transglycosylation reaction. And the bactoprenol monophosphate is recycled here. So very similar, and then after the LPS has been formed. There's a whole complicated multiple protein complex which takes the whole thing and moves it out to the outer membrane. So that's how it works. Okay, outside of the cell wall, bacteria can have a capsule antigen. So you can kind of see the capsule on light microscopy, this halo around the bacterial cells here. And by electron microscopy, sometimes you can see the same kind of thing. It's a kind of what's noted as a glycocalyx here. So the capsule is a polymer, mostly polysaccharide, but sometimes can be polypeptide. For example, Bacillus anthracis has a polypeptide capsule. And it can be present in gram-positives and gram-negatives. But it's a facultative structure, okay? Not all bacteria need a capsule. And within a single species, you can have encapsulated and bacteria without the capsule. Oh, yeah, that's what I just said. Now, okay, because antigenically, these capsule antigens are variable between species and within a species, so they're useful for identification, and this is called a K antigen. K for capsule in German, I guess. And its function is to protect, it's kind of like LPS, to provide another protective layer on the outside of the bacterium. Can also be useful to protect against attack by bacteriophages, viruses that infect and kill bacteria. So if there's a big solid capsule around the outside of the bacterium, a virus that's going to come from here and is trying to fix on a receptor on the outside of the cell wall won't have access. So it's a mechanism to protect against bacteriophages. Can also be useful to retain water. So if the bacteria are left out on a surface, then the capsule antigen and the biofilm can retain water and keep the bacteria alive. And in terms of pathology, a major role of the capsule antigen is to inhibit phagocytosis of bacteria by neutrophils and macrophages, cells of the immune system that can internalize and kill bacteria. And this is a major virulence factor for several bacterial infections. So Streptococcus pneumoniae, this is the big classic. Strains that have a capsule can be pathogenic. Strains that have no capsule are not pathogenic. How is this thing synthesized? Same kind of problem as for peptidoglycan and for LPS, the same solution. So once again, what you have is synthesis of the repeating subunit on bactoprenol. It's going to be flipped in the membrane, and then this subunit is transferred by transglycosidase onto the growing chain. And then finally, the whole thing's transported to the outside of the cell. So for this, and for the others, you don't need to remember WZC, WZ8. It's not important, but I think the reason I want to show you this is to get you to understand that once you know and you understand how peptidoglycan is synthesized, then it's the same kind of mechanism for the synthesis of these other important structures on the outside of the bacterial cell. Is it just me, or is it kind of getting warm in here? No, do you want to open it? No? Just me, okay. I'm going to close this window because the light is... Okay, that's fine. Okay, so Fimbria and Pili. So Now we're going to look at the filamentous structures on the outside of bacteria. So these are two names for the same thing. Okay, so Pili is like a poil, okay, so it's hair. And fimbriae, I guess, is means like threads, they feel. And there are two names for the same structure because you had two microbiologists who observed them and described them independently. So they both made the same discovery and gave them different names. The fimbriae, this was a microbiologist in Dundee, in the northeast of Scotland. Yes. Pili, this was some American person. Okay, so fimbriae and Pili, you can have hundreds of them per cell, very, very narrow, 3 to 10 nanometers in diameter, and they're not all the same. One cell can have several types of fimbriae or Pili on the surface. And because they're not the same, they have multiple different functions. They can be involved in adhesion, either to inert surfaces or to cells in your body. So this is another virulence factor for pathogenic bacteria, expressing an adhesin that allows the bacterium to attach to and colonize the host. Other types of Pili can be secretory. They can secrete proteins outside the cell, including virulence factors, once again, that are going to damage the host. And other types of Pili can allow bacteria to move across a surface. So these are involved in gliding motility. Now, the other type of Pili are F-Pili or conjugation Pili. So they are fewer, most generally 1 or 2 per cell, but maybe several. They're wider in diameter, and they are, of course, essential for conjugation. That's the transfer of DNA from one bacterium to another. So this is striking electron photomicrograph of two bacteria conjugating with each other. So one of these two has shot out an F. pilus, and it's attached to the other cell. What happens next is that this conjugation pilus is going to retract, and the two cells are going to come together. And the DNA transfer occurs when the two cells are very close together. So it's not like you have a kind of connection like this, and then the DNA shoots up all the way over there. This is more like a kind of like a harpoon. It's going to be shot into one bacterium, and then the other one winds it in, and they get close together. Okay. So that's kind of impressive thing for a structure to do. It can extrude and it can be retracted in a controlled way. Now, to understand the different functions of the different types of Pili, I'm going to go through this slide, which I admit is a little bit complicated. So what you need to understand is you've basically got 4 types of Pili. Now you might imagine that microbiologists would call them type 1, type 2, type 3, type 4, right? And initially that was what was attempted by serological techniques. You got antibodies that could react against one type and not against the other type, and there were 4 groups. And then later on, there was molecular characterization of what these types 1, 2, 3, 4 were made up of. And it turned out that type 3 and type 4 corresponded to real objects that resembled each other, but type 1 and type 2 were not valid groups. Okay, so type 1 and type 2 doesn't exist anymore. Type 3 and type 4 still exist. So type 1 and type 2 don't exist. What have they been replaced by? They've been replaced by these 2 different types of Pili, chaperone usher Pili and curly Pili. Oh, yeah, and one thing I should also point out is this is true for gram-negatives. Okay, so these gram-positives have some types of Pili, but they're not so diverse. They have something a little bit like this. They have curly type Pili. Now, once again, I don't think you need to try and learn the different mechanisms of biosynthesis for Cu and curly Pili. The only thing that's important here is that the components of the filament, they're exported into the peroplasm, and then they polymerize in the outer cell membrane for Cu Pili, or they're exported outside the cell and add on to the extremity of the pilus for curly. So the biosynthesis occurs on the outer membrane or outside the cell. Now, what does that mean? That means that it's irreversible, because you can't really, the bacteria can't really control what's going on here. Okay. So once chaperone, usher, or curly Pili have been synthesized, they're on the outside of the cell, they stay there. And they have very simple functions. They're basically only used for adhesion. So all this other clever stuff, secretion, retraction and everything, that's the other types of Pili. So let's have a look at type 3. Okay, so type 3 Pili. They're formed by a big multimolecular protein complex that that crosses the whole cell wall. So there's part of it anchored in the outer membrane, part of it anchored in the plasma membrane, and it crosses the peptidoglycan layer. So the filament proteins, here it's called PRGI, synthesized on the ribosome, and then they're transported inside through the filament, which is hollow, and they're added onto the tip. So these Pili grow from the tip, not from the base. Now, because this is a hollow tube, which is not the case for these filaments are thin, and there's no space in the middle. Whereas for type 3 and type 4 secretion Pili, this is a tube. Okay? So they are used for secretion. So Salmonella typhimurium, type 3 secretion Pili, is used to secrete virulence factors and, in fact, inject bacterial proteins into host cells and kill them. Okay, so the function of type 3 Pili is secretion, not really adhesion, not motility. Type 4 Pili, let's have a look at this one. So once again, they're formed by a big multi-protein structure that crosses the cell wall. But this time, the subunits that make up the filament, Pili, they're added onto the base. So the polymerization is at the base of the filament. So new subunits are added at the base, and this pushes the whole thing up. Okay? That's very important, because this process can be reversed. Okay? So that means that, okay, when filaments are getting added, when filament proteins are added, the filament's going to extrude from the cell. When filaments are taken away from the base, it's going to come back in. It's going to be retracted. And that means, okay, so type 4 Pili, retraction is possible. And their function is in adhesion and gliding motility. So how does that work? Well, if you've got bacteria with type 4 Pili that are on the surface, they can extrude type 4 Pili out in this direction. They'll bind here through their adhesion function, and then they can be retracted. So they get shorter, and this bacterium is going to start moving in this direction. So you've got you can kind of imagine it physically. You know, you're throwing out some kind of rope, and then you pull it in like this, and you move forward. You throw out a bit more, and you pull it in. So this is gliding motility, bacteria moving across a surface. It can only type 4 Pili can do this. Type 1, okay, curly, CU, and type 3 can't do it because they can't be extended and retracted. And that leads us to the kind of last type, which is type 4 secretion Pili. So this is type 4 Pili, can be extended, can be extruded, can be retracted, but they're hollow and they can be used to secrete proteins or maybe DNA. So in fact, these ones, conjugation Pili, they're a modified form of type 4 secretion Pili. Okay? Because they've got the function extrusion, retraction, and secretion. Okay, is that okay for everybody? I know it's a bit complicated. Okay, so the other type of filaments that are on the outside of bacteria are flagella, whips, and you can have one, several on one end of the bacterium or several all the way around, and in that case you can characterize them as monotricious, lophotricious, or peritricious, and that's useful for identifying bacteria. And when you look at the structure of the bacterial flagellum, you have one of these multi-protein complexes that spans the cell wall attached to a big long filament. And the way it works is that at the base plate here, this is kind of like an axle, and this is a molecular motor. So when protons translocate across the plasma membrane, it causes this motor to turn round. And it can run in two directions, like this or like this. So these are kind of the biggest types of filament on the outside of a bacteria, the biggest, largest in diameter and the longest. So this is longer than a bacterial cell. Bacterial cell can be, I don't know, 2 or 3 microns long. The flagellum is like 5 times longer. It's quite big. Now, all of this filament is formed by hundreds, thousands of copies of the same major filament protein, and this is what constitutes the H antigen on some gram-negative. So it's useful, once again, for identifying bacteria. So facultative structure, not all bacteria have them, and within a species, some can be flagellated, some not, and the antigens can be different. So if you see something like this, H70157, H is the serotype of the flagellar antigens, O is for the LPS, right? So H is because these antigens are sensitive to heat. They're made of protein, they can be denatured, whereas the capsule antigens and the LPS antigens, they're polysaccharides, they resist heat. Okay, so the function of flagella is to move the bacteria around in liquid. Okay, so type 4 Pili can be involved in mobility on a surface. Flagella are for moving around in a liquid. And basically, if this motor here starts turning round, the whole flagellum is going to start spinning, and this will generate force to push the bacterium around. Now the question is, before that, okay, before doing this, okay. Before that question, there's another question, right? So another aspect. So, okay, how does the major filament formed? Well, in fact, subunits are produced on the ribosome. They're not in a chain like this. This is a kind of mistake. So you just have individual subunits produced, and then they're transported up through the inside of the filament, and they attach on the end. And that's how it elongates. So what does this remind you of? Okay, I can see that on your faces that the answer is nothing. It's got a the filament proteins are added on the end. They're transported up the inside of the filament. It's one of these, right? Yeah, type 3. That's exactly what it is. So in fact, a lot of the base proteins here on the basal part of the flagellum, they're similar, they're homologous to a type 3 secretion system. So in fact, this is a complicated molecular machine. But it didn't evolve out of nothing. Once you've got a type 3 secretion system, you just got to, stick some motor proteins around the end of it, and there you go. You've got your flagellum. You can move around. You don't have to build it out of nothing. You just add something on to a pre-existing structure. So that's the point of that. Okay, so how do bacteria move around in a coordinated way with flagellar rotation? So kind of if you or me were trying to find a way to move about in 3 dimensions, you might think, okay, we need 3 kind of motors, okay, one for forward and back, one for left and right, and one for up and down. And if we do that, we can move around in 3 dimensions. That makes sense to me anyway. But bacteria aren't like that. Okay, flagella, they can't move up and down or from side to side or or forwards and backwards. They've only got spinning in one direction or spinning in the other direction. Okay, they've got clockwise and anti-clockwise. And depending on the sense of the rotation, this will produce 2 different types of motility in bacteria. So if the flagella rotor is turning around anti-clockwise, this causes the bacterium to kind of move ahead in a straight line. And if it's turning in a clockwise sense, then the bacteria will start to tumble around randomly. And that's true for monotricious, peritricious, and what's the other one? Lophotricious flagella. Okay, so for this kind of peritricious bacterium, multiple flagella all over the surface of the cell, if they're turning in an anti-clockwise sense, they'll all be coordinated together. So the bacterium moves forward. If they turn around in the clockwise sense, they're all going to be turning in an uncoordinated way. So the bacteria is going to move around randomly. So instead of having forwards, backwards, left, right, up and down, bacteria have got forwards and random. Okay, those are the 2 directions of motion for bacteria. And combining these 2 modes of movement allow the bacteria to, or rather switching between them, allow the bacteria to move towards things that bacteria like, like sugar. They're like children, really, right? And move away from things that they don't like acid, areas with too much oxygen or not enough oxygen, depending on their physiology. So how does that work? Okay. So imagine you've got some sugar around here, high concentration of sugar somewhere, a little blob of organic material floating in the sea, and it's giving off some sugar. So there's going to be a concentration gradient of this sugar. It's kind of like this. The closer you get to the source, the more there is. So you've got to imagine you've got bacterium floating around here with its flagellum. And let's imagine that it's turning in an anti-clockwise, it's rotating in an anti-clockwise direction. So the motion is going to be forward. It's going to shoot off over here somewhere. So in fact, it's going to go down the concentration gradient. And somehow in here there's going to be a little regulatory circuit that detects we're going away from the sugar. And this has a consequence of changing the flagella rotation to clockwise. So this bacterium is going to start tumbling, going to start moving in all directions. And then after a while, it's going to switch to anti-clockwise rotation again. So it might end up pointing kind of this way with anti-clockwise rotation, and then it's going to carry on moving. So as long as the bacterium is moving up the concentration gradient, the sense of rotation is not going to change. And it's going to carry on moving, moving, moving until it goes too far. Right? Once it starts going down the concentration gradient again, then we'll have another switch. So it's going to start moving in a clockwise rotation. It's going to be random. So you have this switch between random and moving straight. So it's going to get closer and closer and closer to the source of the sugar, just by a very simple algorithm of, if I go too far, I better change direction. I'll change direction randomly. If it's the wrong direction, I'll change again because I'll be going in the wrong direction. And I'll keep going the same way as long as I'm getting more sugar. Okay? So very simple mechanism, okay? Forward and random, and I'll switch between them to go towards things I like and go away from things I don't like. Okay. Right, so that's about halfway through. So just to make sure everybody's been listening, so think about this for a couple of minutes, and I'll ask everybody what the answers are. Okay, so which of these are useful for identifying bacteria? The LPS core oligosaccharide. Who says yes? A couple of people. Who says no? Some more people, a lot of people not really taking any risks, huh? It's okay. There's no punishment for getting the wrong answer. So the answer is no. It's too conserved, okay? It's a conserved structure among all gram negatives. So in fact, you're not going to get any useful information from this. LPS O repeats, that's useful. Who says yes? Who says no? Yes, the answer is yes, okay? It's variable between species, variable between strains. CU Pili, that's going to be useful or not useful? Who says useful? Who says not useful? About 50-50 with a small sample size. The answer is not really useful because they are facultative, they're often not present, and whether they are present or not doesn't really correspond with a particular strain of bacterium. So they're too variable to be useful, in fact. Okay, so core oligosaccharide, not variable enough. CU Pili, too variable. Capsule antigens, useful or not useful? Who said yes? Yeah, you're right, very useful. Peptidoglycan, useful, not useful. Yeah, you're right, it's not useful. Why? Why isn't it any use to distinguish between bacteria, peptidoglycan? Yeah, same thing. Yeah, it's too highly conserved. There are no differences between bacterial species. Okay, so we're finished for the outside of bacteria. Now we're going to go inside. It's not very complicated inside bacteria, to be honest. Very, very simple. So the only real kind of internal structures they have are inclusion bodies, which are there for storing any kind of nutrient that bacteria need in relatively large quantities for their energy metabolism. So that can be the main elements necessary for life: carbon, nitrogen, phosphorus, and sulfur. Okay, they're needed for synthesis of all sorts of biomolecules. So for carbon and nitrogen storage, bacteria can produce granules of glycogen, or kind of droplets of, granules of PBH, which is polybeta hydroxybutyrate, or PHB rather. Cyanophycin, so this is a polymer of arginine and aspartate, so that's a storage for storage of nitrogen and carbon. And for inorganic molecules, polyphosphate sometimes can be stored in granules, and sulfur for some bacteria which use sulfur as the basis of their energy metabolism. So these purple sulfur bacteria, which are photosynthetic and they use reduced sulfur as a source of electrons, they can stock granules of molecular sulfur inside the cell. So these granules can either be directly in contact with the cytosol if the polymer is hydrophilic. So that's the case for glycogen, cyanophycin, and polyphosphate. So these can be directly in contact with the cytosol. But hydrophobic polymers can have a protein or lipoprotein shell around them. So that's true for PHB and for sulfur. So we're just kind of as an aside. So you can identify these PHB granules in transmission electron microscopy, because during the preparation of the sample, there's going to be a wash with something like methanol or chloroform, and this will totally dissolve the PHB. So that's why this looks like empty, because all of the polymer has been washed away. Okay, the nucleoid. So this is where the bacterial DNA is. It's concentrated kind of in the middle of the cell, and it's condensed. How do we know it's condensed? Because if you get bacterium and lyse it very gently, treat it with proteases to destroy all the proteins, then the chromosome can leak out and expand and come out of the cell, which you can see here. So just like inside our cells, the DNA molecule is far too long, far too big to be inside the cell if it's in an extended conformation. So the bacterial chromosome is condensed, and it's associated with polyamines like spermidine and a dimeric protein called HU. So the HU has a function of introducing curvature in compacted DNA structures. So the DNA spermidine is kind of compacted, and the HU allows it to bend around so that you can have the structure turned in on itself. So some archaea have histones instead of for just like eukaryotic cells. And there is one weird exception, which are planktomycetes. This strange thing, which was kind of a big surprise when it was discovered about 20 years ago, is that this bacterium has actually got all of its DNA inside a membrane-bound structure. So it's kind of like a prototypic nucleus here. Kind of very, very unusual. Okay, cytoskeleton. So as I said the other day, perhaps, for a long time it was thought that bacteria do not have any cytoskeleton at all and didn't have homologs of the major cytoskeletal proteins actin and tubulin. But in fact, that's not entirely true, because many bacteria have structural homologs of actin, which are called MREB in gram-negatives and MBL, so that's MREB-like in gram-positives. And so you can get many copies, multiple copies of MREB, polymerize together to form filaments. And these filaments are tagged with GFP in this image here. You can see that it forms a kind of like helix. And the function is to give a kind of elongated structure to the cell. So if you knock out MREB from the genome of these bacteria, then they become spherical. So the presence of MREB and MBL is what gives you the form of bacilli. Okay? So this is the difference between cookie and bacilli. Bacilli have got MREB or MBL. Now what about tubulin? There is one structural homologue of bacteria of gamma tubulin in bacteria, which is called FTSZ. And yeah, like eukaryotic tubulin, it can exist in a monomeric form in the cytosol, or it can polymerize into a ring. And the role of FTSZ is essential for correct cell division in bacteria. So as in growing bacteria, then you'll have FTSZ in a monomeric form. But then as DNA replication starts and the two daughter molecules of the chromosome begin to separate, then FTSZ will begin to polymerize and form a ring around the middle of this growing bacterial cell. And the ring formed by FTSZ is what is going to initiate the scission or the separation of this growing cell into two daughter cells. So apparently FTSZ here is not like some kind of muscle which is going to close around the middle of the bacterium and chop it in half, but it's a scaffold which is going to organize all the other proteins that are going to be important in, cutting the cell wall in two, making the membrane break up in two from the plasma membrane. So FTSZ is has an essential function in separating two bacterial chromosomes during cell division. Personally, I find this really interesting because if you think about mitosis, you have the mitochondrial organizing centers formed from gamma tubulin, right? And they're essential to organize the mitotic spindle, which is how the daughter chromosomes separate during mitosis. So even though mitosis and bacterial DNA replication are very, very different processes with huge differences, you've got kind of at the center of how these types of cells are going to separate the chromosomes, you've got this tubulin-like protein which is at the middle of it all. So it's really suggestive that sometime way, 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 way back, there's going to be a common origin of these processes. So kind of what I'm trying to say here is that, when you look closely at bacteria, you get the idea that they have a very, very simple version of a lot of what goes on in eukaryotic cells. They've got a very, very simple cytoskeleton. They've got a very, very simple way for keeping DNA compacted inside the cell. They've got a kind of very, very simple kind of organization of what's inside the cell. But it's not like nothing, okay? There's a kind of simple precursor for things that are going to be very much more complicated in eukaryotes. Okay, we're going along, in fact, far too fast. So apparently we have half an hour to think about bacterial endospores. Let's say maybe 20 minutes, and then we'll have a break. So endospores are formed by some gram-positive bacteria, never in gram-negatives. There are no spore-forming gram-negatives. And the position of the endospore within the bacterial cell is a kind of criteria for identification. So you can distinguish bacillus from clostridium because of the position of the spore. Endospore, they're found inside another bacterial cell, vegetative cell. So I guess, yeah, first I'll talk about the structure of the endospore, then about how spores are generated, and then how spores are reactivated. Okay. So let's think about the structure first. So going from the outside to the inside, often there's an exosporium, so a loosely attached coat of glycoprotein on the outside. And then you've got a very, very thick spore coat, which is formed mainly of protein, which is very, very heat resistant. These proteins have got a lot of disulfide bonds, so the structures are of the secondary structure of the proteins are highly resistant to heat. Inside that, in the cortex, this is a thick layer of peptidoglycan associated with this small molecule, calcium dipicolinate. Then you've got the spore wall, peptidoglycan wall, and then inside you've got a dehydrated cytoplasm. It's got ribosomes, okay, the bacterial chromosome. It's got DNA repair enzymes and SASPs, small acid-soluble proteins. So acid-soluble, these are small basic proteins, and they have a role in compacting the DNA. So what is the function of all this? Well, the function is this is a kind of dormant bacterial cell which can resist very, very harsh conditions. It's very resistant to heat. It's very resistant to chemical inactivation by things like bleach, acid, because all the chemical agents are just going to penetrate up to here and bind to the proteins here, but won't be able to get to the DNA in the center of the spore. So it's a very, very tough, resistant, dormant form of the bacterial cell. And they are produced in response to poor growth conditions for bacteria. So often sporulation is induced by starvation. You grow bacteria up in a rich medium, then put them into a medium with nothing. They'll start getting hungry. They realize we can't survive if we carry on growing, and sporulate, produce spores. Okay, it's not the only inducer of sporulation. One other example is Bacillus anthracis, anthrax bacillus. This is an anaerobe. Okay, so it likes to grow inside infected animals, infected people. And when that animal is sick and will start spitting out infected saliva and blood, the bacterium finds itself out in the air in the presence of oxygen. And it's oxygen which will induce sporulation, because it's a signal for the bacterium that, I'm outside of my host. I've got to sporulate, produce spores, and just remain dormant in the ground. So sporulation is a response to poor growth conditions. Okay, but if bacteria are going to become dormant in the form of an endospore, they have to wake up another time, right? There's no point being dormant forever. Just the same thing as being dead. So endospores can be reactivated and germinate. And okay, when's a good condition to start growing again? When you've got food, right? Good time. So the presence of peptides and sugar is often something that can reactivate spores, but on their own, often not enough. Often there needs to be a kind of heat step to reactivate spores, especially with some moisture. So what seems to happen is that the first step is heating 65 degrees in a kind of moist atmosphere will allow some rehydration of this outer spore coat, make it a little bit more permeable. And then if you've got peptides or sugar molecules, they can diffuse in here and get to the cortex. So once they're in here, they'll stimulate the release of the dipicolinate, which will diffuse out again. And this will activate enzymes that start to degrade the spore coat. So this is the function of the dipicolinate. It's a signaling molecule. which is going to be released during germination, activate enzymes to degrade the spore coat, and then this will allow the bacterium to have more access to water to rehydrate the cytoplasm, sugar, peptides, whatever is outside, then the metabolism can start up again. DNA repair enzymes start getting to work, repair any damage to DNA, because this spore might have been dormant for several years. Even if it's very resistant, you could have, some UV radiation coming to damage DNA from time to time. And then the bacterium will start to grow. So those are the events inducing sporulation and inducing germination. Now, another question here is, how on earth can this tiny, metabolically inactive spore produce all of these big, complicated, thick structures on the outside of the endospore? And the answer is, well, it doesn't. During sporulation, what is going to happen is that initially, so in a rapidly growing cell, this is in like bacillus here. Your culture of bacillus has got plenty of sugar, plenty of peptides there, it's growing happily. So during cell division, each daughter cell is going to be identical to the next one. So the bacterial culture doubles. During sporulation, that's not what happens. You have an asymmetric cell division. One daughter cell is going to be big, and the other one is going to be smaller. And the destiny of these two cells is going to be very different, because the larger daughter cell is going to engulf the smaller one, and at this point, the small one is going to become the spore. So it's going to shut down its metabolism, shut off the expression of most of its genes, and just retain these few components that you find in the endospore. Whereas the larger daughter cell, which has engulfed the smaller one, is going to synthesize the different layers of the endospore coat. First, the cortex, then the coat proteins, and then finally the exosporium. So it's the large cell which is around the endospore which does all the work, which does all the synthesis. Then at the end of the process, this large daughter cell will die. It lyses. So here, once again, you've got a very simple example of something which is present in eukaryotes and especially in all multicellular organisms. You have differentiation into two different types from one mother cell. And you've got programmed cell death. The cell type that's not needed is going to be deleted. These are absolutely essential for differentiation and producing complex multicellular organisms, having different programs of gene expression for differentiating into types of cells that have different functions. So you've got a kind of very, very simple micro version of this in bacteria. So it's not the only example. You have types of bacteria that can grow into kind of elongated colonies with maybe 2 or 3 different cell types. And endospore formation is, perhaps the simplest example of cell differentiation anywhere in biology. So once again, we've got this very, very simple version of a process that's essential for eukaryotes in bacteria. Okay, one question, and then we'll have a little break, I guess. So I don't want to get anybody hungry, but just think about this for a couple of minutes.